Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Neurogenetics. Neurogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hello, everyone. I'm very excited to be here today with Dr. Andrea Gropman, and we're going to explore the impact of genetics and genomics in modern medical practice today. We're going to go from the bench to the bedside and really understand what are both the opportunities but also the challenges with taking some of the most exciting technologies that are being developed right now and actually getting them into the healthcare system to have an impact on patients. If you're not aware of Dr. Gropman's work, she's done a ton of amazing work in both pediatric neurology, genetics, and a lot of work in inborn errors of metabolism. So you're going to learn about some new areas of biology if you're not familiar with these already, but also hopefully give you a lens of what it takes to bring some of these new cutting edge technologies that actually have an impact on patients every day. So Andrea, thank you so much for taking the time to join us.
Thanks for having me today.
My pleasure. I would love if maybe you could start with a quick background on yourself. What is the kind of work that you do today? What really gets you excited? And a little bit about how you got into this field in the first place as well, I think would be really interesting for people.
Yeah, sure. So I am multiple trained across different specialties. I'm a child neurologist, a clinical and biochemical geneticist, and a developmental pediatrician. And most of my research has focused on using non-invasive imaging technologies to establish biomarkers for several of the inborn errors of metabolism. Most of my work, however, has been in urea cycle disorders using multimodal MRI, so different types of MRI. So structural MRI, functional MRI, which now we've replaced with functional near-infrared spectroscopy, which is a wearable device. So that came about due to COVID and trying to look for ways to innovate our research and out of necessity because of the scanners being closed. And then I also use magnetic resonance spectroscopy, also doing some bench to bedside precision therapy for mitochondrial diseases with drug repurposing, a lot of teaching of the next generation, mentoring. And so I got into this, I guess, really in medical school. I was really interested in genetics, but the field wasn't fully developed. And I always had a background interest in neurology. I'd worked in labs that focused on neurologic disorders. And then as time went on, just seeing the real connection between, neurologic disorders that had genetic underpinnings, either from getting family histories or getting actual results. I got really interested in the inborn errors. I was a biochemistry major in college, so that really piqued my interest that there was actually a field where you could apply that knowledge. And then when genetics became more of an actual separate field, and it looked like things were going to really grow and grow, I decided after my neurology training to go back and train also in genetics, and in my work today, just combining the knowledge of across the two fields to investigate these disorders, to develop biomarkers, and to take care of patients.
Amazing. So if we focus on the patients and the families, I guess, to start with, what kinds of families do you mainly work with that you see, and at what point in their journey do they come to you? Maybe you could walk through an example or two, blinded, of course, and feel free to mix a few together. So we're not talking about any individual case, but I think it'd be good to get it for people to get an understanding of actually what does a patient or family that steps into your office look like? What kinds of problems are they experiencing, and what is that journey like for them?
Yeah, so, because I've been doing this for 25 years or so, things have sort of streamlined. So whereas in the beginning, I saw all kinds of families, now I've really focused on those that are also linked to my basic research endeavors. So I see a lot of patients with urea cycle disorders. I work with my genetics colleagues, see them in multidisciplinary clinics, but mainly focusing on the neurologic aspects of the disorders. I see many known and rule out mitochondrial disorders, both pediatric and adult. And then by nature, the fact that I was already trained in neurology when I started my genetics fellowship, and this was at NIH, I got pulled into different disorders that then also became side interests. So for example, Smith-Magenis syndrome, which is both a chromosome deletion as well as a single gene mutation within that deletion. The XY chromosome aneuploidies was almost a hobby with a colleague of mine, and then has sort of grown into multiple clinics across the world. So I've had the opportunity to see patients with those conditions, not only in the US, but also the UK and Italy and Australia. So that's really grown out as well. And then I think other patients in whom they just don't have an answer. Like I seem to be the go-to person for when the testing has been exhausted, or, all the great minds have come together, but they haven't really thought or, found the cure for the problem, and they don't know what the answer is. I did some work, and I still continue in the undiagnosed diseases program at NIH. So I see that as an extension of that type of work. So I think leading to the topics you may discuss often have to go back to the basic science or the bench to answer these questions that the clinical studies don't really provide answers for.
Yeah, I'm interested in starting with this diagnostic question, I guess. How much in the last 25 years has genetics and genomics changed that equation, in a sense of the number of patients that come to you or come to the broader clinics that you're a part of that actually do get an answer now and allow you to do maybe some more helpful things. But we should after that talk about the percentage of people who still don't get an answer despite next generation sequencing. But I'm interested in that change. How have you seen that? How's that affected you? And what learnings maybe could we take from the improvement in diagnostic yield that hopefully you've seen as a result of this over the last decade or two?
Well, it's changed significantly. And I think those that are just trained, the newly trained younger physicians, don't have an appreciation for the fact that 25 years ago, what did I have at my disposal? I had a karyotype pretty much.
Yeah.
Fish was coming online. We had the back arrays, which missed whole portions of chromosomes. So we were very limited. We knew our patients had, for example, some of our dysmorphic patients had probably a deletion, but we couldn't find it because of the coverage. And then that got refined. And then, of course, the first generations of whole exome. We had some panels before that. I remember in the mitochondrial field, which is something that I'm very interested in, we had a panel of mitochondrial DNA that maybe had 5 genes total. That's it. So we were doing a lot of muscle biopsies back then or other studies to try to determine if someone had a mitochondrial disease. So we didn't really have much. And then, so taking the family history and the history and exam were very important, as well as MRI. But, also MRI evolved. That's a whole separate issue to discuss as well. But, then we started having our first, exome panels and our exome sequencing rather. And then, that became refined, and now having whole genome panels improving. So really there's been a vast increase. So I'd say that the likelihood of diagnosing some of these patients who previously weren't diagnosed increased, but we're not at 100% of those that we suspect. And also more recently recognizing the possibility of a dual diagnosis. So we were all taught, oh, all the symptoms must neatly fit under one diagnosis, just, kind of crush them in there and make it fit. But then we realized as we were testing patients and finding a result that that particular gene variant may only account for part of the phenotype, but not all, and that it's necessary in those cases to do additional testing. And there may be a dual diagnosis that is found by either doing, if you started with an exome and then you did a microarray, or you went on to do a whole genome, that then there's a second variant that explains the other part of the phenotype. So I think dual diagnosis is really something that we need to think about as well, and to push, to do additional testing if the patient's phenotype doesn't fit completely even within the spectrum that we see with certain single gene disorders.
Yeah, absolutely. So would you do something like whole genome sequencing as standard now? And how does insurance coverage or those kind of things play into it? What's the approach there?
So I'd say it depends, and it depends very much on insurance coverage. So we cover the District of Columbia, Maryland, and Virginia patients, and they all have different rules with regard to Medicaid, which is the public assistance, and private insurance, and the military insurance, TRICARE. They have their own insurance. So I think in the moment when we see the patient, if we're able to do either a whole exome or a whole genome based on insurance, and in DC and Maryland, so far, I mean, these could definitely change because this wasn't how it was 10 years ago. They will cover the cost entirely. So we can do the testing in the clinic, or at least send the kid home. So we may decide, we might want to do the whole genome. It's case by case. For someone who has private commercial insurance or has military insurance, we have to go through a prior authorization process. So we may be told what we can do, even though it may not be the test we want. And I know in the past, maybe 15 years ago, TRICARE was not approving Fragile X, for example, which we still have to remember to test that unless we do the genome, which includes, can test for the trinucleotide repeat. So a lot of it is driven by the insurance. And then it gets worse on the adult end. Like, I had to laugh. I subscribe to the neurology journals, and they have this neurology review that I'm looking at, and they talk about genetic testing is recommended for adult patients with epilepsy, and that there's a false misconception that genetic testing should only apply to children. It's like a decade, overdue. It's like finally, they're
Right.
realizing it. So I think, but it is hard to get insurance authorization for adults in particular. And we do see many late onset of the inborn errors of metabolism in adults presenting, and we don't necessarily have the means to test them. So I think that's a big gap in our payer system here in the US, is that adults, can have genetic disorders that present in adulthood, or might have had their seeds in childhood that maybe the symptoms were mild or overlooked.
Yeah, that's interesting. You mentioned you spent some of your time teaching and mentoring. How much of that is working its way into medical education? Do you think we have hope for the next generation that they'll grow up understanding that genetics is a lot more complicated than the textbooks imply, and we have a lot of these issues, or is there still some work to be done at maybe at the medical school or residency level in terms of how we do these things?
Yeah, there's definitely more to do. I remember, years ago teaching genetics at Georgetown, and the total was a 2-week block. And
Yeah.
I think at, the other medical schools we're affiliated with, it's a very small block. And just by being on inpatient rounds and working with the residents in the clinic, the knowledge is slim. They don't really get it. I don't know. Some medical schools may be better than others. There's a lot of variability. I think we had way back when there really wasn't even anything to talk about. We had a pretty decent genetics block, which is how I got interested in it. I don't think I would have been as interested if it was just a 2-week block, but It was comparable to the time we would spend on the other specialties. But I know that's not the case everywhere. So, and about maybe, I don't know, 20 years ago, I put together this conference called Genetics for Your Practice. And again, to just show that genetics was going to permeate different areas: pediatric, adult neurology, neonatology, cardiology. And I think that's really become the case, is that there are, at least at my hospital, and I know many other children's hospitals have genetic counselors within other subspecialty clinics where there's a high likelihood of genetic disease, like cardiology, like endocrinology, for example, obesity, orthopedics, perhaps, other specialties, cancer, where there is a genetic counselor. And so genetics is its own specialty, but it's also embedded within other specialties. And I think that will evolve over the future, whether genetics is going to stay separate or it's just going to be taken into the other specialties to a larger degree.
Yeah, what's your instinct on that? Is it going to be taken into and just permeate every I guess existing specialty, or is it going to go the other way and become more and more a specialty in its own right?
Well, it is already a specialty in its own right, and I think it will stay that way, especially for the inborn errors, because nobody wants that. They're too complicated and they require a lot when a sick patient comes in. But I think, at least from the counselor standpoint, we may see more genetic counselors within other subspecialties where testing may be required, genetic testing. But I still see genetics as being a strong specialty unto itself, because there are many conditions that probably would not be infiltrated into other specialties or that need to have that expertise stay local.
Yeah, that makes sense. I was curious to learn more about imaging, actually. You mentioned this at the very start, and it sounds like you've done some really interesting work looking at a lot of different imaging technologies. Maybe you could talk about the role that plays. My assumption is that we're talking now after a diagnosis is made, either after a diagnosis is made and you're trying to understand a little bit more about functionally what's going on, or maybe it's also a useful diagnostic tool in its own right. I'd love to hear a little bit more about how the role that it plays in the way you deliver care, but also some of the technologies you talk through. I think it'd be interesting to understand that evolution to more and more wearable and remote-based technologies, which is pretty interesting.
Yeah, sure. So when I first got involved with the inborn errors with the urea cycle in particular, there was actually an opportunity. I think a lot of us get where we are in our fields just based on chance and opportunities. So there was an opportunity to train in neuroimaging, multimodal neuroimaging, and I wanted to apply it to the inborn errors. So, of course, the first disorder I was working on were the urea cycle disorders. And so, there's an opportunity there, because naively I thought, oh, the first project I'll do is just collect all the MRIs and see what all the changes are. But even though the patients present in coma, nobody ever imaged them. They just assumed that all of the neurologic features were due to, in the case of some of the urea cycle disorders, a high ammonia level in the brain. And that they knew way back when, before we had the treatments we have now, that the patients would have, moderate to severe neurologic complications, and severe in some cases. And that was just where the bar was. It was very low, that either you didn't survive or you survived with major deficits. But nobody really knew, what was going on inside the brain or why. So, I think I had probably the first not the first, but the largest study of patients who actually were stable, imaging them from multiple perspectives, because there were so many aspects of the impact of hyperammonemia that one type of imaging modality just wasn't going to capture it. So, for example, many of the patients don't have changes right away on just a regular T1, T2 MRI, or they do when it's in specific areas. But, luckily for us, using magnetic resonance spectroscopy, which looks at chemicals in the brain, 3 of the chemicals that we can measure are actually altered in urea cycle disorder. So one is glutamine, which is your surrogate for ammonia. And so when your glutamine's high, it's an indication that your brain ammonia load is high, and that can be different from what's happening peripherally in the body. So because they're 2 separate boxes and they don't necessarily connect, is that you could have a normal ammonia in your blood, but have an elevated glutamine or surrogate for ammonia in your brain. The other 2 are myoinositol, which has an inverse relationship to ammonia. So that's your buffer, if you will, for when ammonia enters the brain, it causes swelling. So that's your stopgap for swelling. Those levels go down. And then in the long term, you have a decrease in choline, which indicates that there's loss of integrity in your fibers, in your white matter fibers. So spectroscopy has been useful also for diagnostics when the MRI is not quite, the T1, T2 is not abnormal. We actually published a case where we made the diagnosis like solely based on the history and the spectroscopy, and gave treatment. And then later the genetic testing came back and confirmed it. And the other one is the diffusion tensor to look at the white matter tracks. And so in one of the conditions, arginase deficiency, which presents like a cerebral palsy, we saw that the fiber tracks going to the legs were decreased. But also interestingly, we were able to look at a patient long-term after recovery from a hyperammonemic episode. And he also had lost some fiber tracks in the legs, which was a new finding for us that we didn't expect. And he had ornithine transcarbamylase. And then everything shut down with the pandemic. And so we needed to look at other ways to look at the patient's brain. And also we were using functional MRI, I should say, as well, to look at some of the cognitive changes, because executive function had been reported as being a major, consequence, even with improvements in treatment and patients surviving and doing a lot better. They still complained of working memory issues and attention. And so we're using fMRI to confirm that, but then the world shut down. So I had colleagues at NIH who were designing functional near-infrared spectroscopy devices. You can get them commercially. The first ones we used were actually ones they had designed in the lab and only covered the forehead. But you can use optodes of light. And we showed that you could get the same type of functional maps as you could with functional MRI. So now we had an option outside the scanner. People could wear it. They could walk around with it. It didn't, need you to be completely still. You could do it on infants. And so we started a lot with that. We expanded that to other inborn errors. And then I think most recently, we are looking at driving. So a lot of our female carriers of OTC had indicated difficulties with driving and attention and visual perception. So we actually put together a driving simulator. Initially, we wanted to try to do it in the scanner, but of course, with all the metal parts, it was just going to be really hard to have the right components. And so we built it outside the scanner, and we're using our FNIRS for that. But we are looking at more wearable devices to look at activity. And then just lastly, you talked about using it for diagnosis before. So we're looking at neuromonitoring. So we got a device called the Moberg, which it's basically a monitor that allows you to plug in every type of contraption that you have in an ICU setting, including an EEG, because my concern was that some of these babies with high ammonia may also be having subclinical seizures, meaning that would only be picked up on EEG, but that they would not be visibly shaking, and that, the outcome could definitely be impacted by whether they were having, seizures during that time. And in fact, they are. And so we have this monitor, and we have some background features on the EEG that we're using as biomarkers to do a bedside precision treatment. So if we see a certain change on the EEG, we alert the geneticists, and they can make a treatment in real time rather than just following a protocol that may say, check this every hour, give this drug, at this. So that's the latest, is we're trying to, really do a precision therapy at the bedside.
That is very cool. I'm interested in newborn screening and how you see this potential paradigm shift impacting what you do, because I would suspect that at the moment you need, when patients become symptomatic, you order genetic testing, and it can take some time, but there's a lot of groups that are working on healthy whole genome based newborn screening that I think could probably pick these up earlier. How much do you think of that? How much do you get involved with those kind of programs, and what do you see as the potential there?
Yeah, definitely a potential for the singleton cases. I mean, if there's a family history already, then we have the variant in that family. We can do prenatal testing and then have a plan for the newborn and how it's going to be handled. And we've done that in a couple of cases where we knew the child may or may not, have the condition and treat them as if until we got the testing back. But having newborn screening or having rapid screening for sick neonates is important. But, one of the questions is, is that too late? Like, is there damage done before? And we still don't know if the first episode represents the subsequent cognitive outcome, or if it's the sequential hyperammonemic episodes, or if it's some combination thereof. So we're trying to use these non-invasive measures to answer that question. Yeah, so, but it would definitely be good to know, and to know quickly, to be able to plan, to stabilize the child, because right now we don't have gene therapy, we don't have a definitive therapy, but some children go on to get a liver transplantation, as do some adults. There have been some adults that have been living with it their whole lives, and then they've decided as adults to get a liver transplant, and it's transformative for them. They can go on a normal diet, they don't have to worry about hyperammonemia. So I think it would be nice, definitely have it. So we are working in the Urea Cycle Consortium. One of our investigators has been working on, newborn screening, and some of them are already on the panels, so we can test, some of these. But, the other thing is some of, especially with the female carriers of OTC, maybe 10 to 15% don't have a variant identified, or they may have what we call private, mutations that are in that family. And then the other question is, what's the prognosis? So, if we do the newborn screening and we find that a child has, for example, a variant for OTC, what does that mean? Is that a bad one? Is that, mild? And so we've been working with Amy Dudley at the Pacific Northwest Research Institute, who on her own, again, serendipity plays a lot into people's careers, I think, was developing yeast models for OTC and looking, sort of as a functional assay for the different variants that have been reported in the patients. And someone that I had worked with on a committee had moved and was in the lab next to her and remembered that I did urea cycle research, and said, hey, you should meet Amy in the lab next door. She's doing something with yeast and OTC. And then, fast forward, Amy's become a collaborator with us in the Urea Cycle Consortia. And so now we have this functional assay that when we do have a variant, we can look and see if she has data from the yeast growth assays to determine if we think it's going to be, a bad prognosis or a milder prognosis. So I think using that hand in hand with a functional assay is really going to be powerful. And she plans to do those assays for the other urea cycle enzymes as well. She's working on that.
Yeah, interesting. And just for people who aren't as familiar with the urea cycle disorders, I think in this case, OTC is ornithine transcarbamylase deficiency,
Yeah.
right? Yeah. So
Yeah.
it's
Sorry. Yep.
is it one of six different enzymes in
Right. So there
urea
are
cycle?
6 enzymes and 2 transporters, so 8 total. And so for most of them, high ammonia is the problem, and it gets into the brain and can cause seizures, and it can cause changes in mental status. It could cause coma if not treated, and even death. And unfortunately, every year we have young adult males with a later onset who present, who nobody thinks about that, and unfortunately they pass away because the treatment and the knowledge isn't there.
Yeah, so OTC is X-linked, I guess, right? Which
That
is
one's X-linked, right? Yeah. Correct. So
females carriers.
Females can be carriers, but many of them are symptomatic. I think we're recognizing, we looked at that also. I'm very interested in carriers of X-linked genetic conditions. And we looked at that. We actually published years ago, on maybe 2010 or 2012, on their cognitive status, because many of them were complaining about attention issues, but because they functioned well in everyday life, nobody really, studied them. And we saw that if you push the envelope and you had them do very hard tasks, that they would start to show some of the features that you would see in the patients who were symptomatic. And then we have something that we're it's under review, looking at female carriers and whether those that aren't symptomatic can become symptomatic later in life. So, so that's an interesting topic in and of itself. Yeah. But yeah, it's so females can be symptomatic. Males, if you have the neonatal onset are symptomatic, and then some males may have milder manifestations and present later in life, even in adulthood, with a trigger. So maybe they've had surgery, or they've had steroids, or they had overindulgence in protein-rich foods. We've seen that as well.
What you when you initially described, obviously we talked a lot about newborns, and I think there's a big, young children. There's a big impact here, but I'm just curious about what we know about the spread of age of onset of this class of diseases. What's currently known about how much of the onset is in childhood versus later in adulthood, and also to what extent is this an open question of we don't actually know how many unaffected, or partially affected adults there are that we just haven't detected because sequencing isn't widespread and the symptoms are difficult to pick up.
Yeah, I would say that we just don't know. Patients may have died and the death certificate may say something else. I think a lot of patients may have realized that they don't tolerate protein and adopted a vegetarian diet and protected themselves from some of the more severe symptoms of hyperammonemia. So I think we just don't know. And I think it's also the common misconception for physicians in general, especially those who take care of adults, is that, genetic disorders all have their onset in childhood. And so they don't think about it. So I think if you don't think about it, you don't do the testing. And if you don't do the testing, then we don't know. And again, when people die and something different's written on the death certificate, did that patient have hyperammonemia due to urea cycle disorder, or, what really was going on? So I think it is an open question. I think, it looks promising now that adult neurologists are going to start testing adults for epilepsy. A little bit too late, but good that they're thinking
Better
of it.
late than never. Yeah.
Yeah, and I can say that because my brother's an adult neurologist. So, so better late than never. So hopefully that type of knowledge gap that's being filled will apply to other conditions where they don't have an answer. And I think you just have to have one patient. So if you have one patient and you test them and you get a positive result, I think that will draw those providers to say, oh, okay, like that was useful, especially if that test influences the care, right? So when we tell patients we want to do genetic testing, it's for several reasons. One is to get that answer, to decrease or to end that diagnostic odyssey. And two is because that answer may have a treatment implication with it. You find something, there may be a drug or a cure or a clinical trial or something that has to be looked at that they're at risk for, and so you change their medical monitoring. And then also the family planning, or who else in the family could be affected. So I think, those are some of the reasons we give to families. So I think, if that becomes more pervasive in the adult population, and we actually did a conference at the American College of Medical Genetics for typical adult neurologic manifestations that ended up being genetic. And I had my colleague present an 82-year-old woman, I think she was 82, to show that, it's never too late. And she ended up having a treatable inborn error of metabolism that improved her quality of life significantly. So you ask, what age is too old? I don't think there is a too old to consider, because sometimes these patients have very complex medical conditions, and especially if it involves different parts of the body, they see this doctor for this thing and, they have a plan, and then they see another doctor and they have a plan for this other part of the body, but they don't communicate.
Yeah.
And try to find a common etiology. They see it as separate conditions because, of course, in adulthood, you can have separate conditions. You can have a heart issue, you can have a kidney issue, you could have, liver issue. They may or may not be related, but they don't think in that same way. I think we do in pediatrics, where we try to figure out, are these all related, or are they true unrelated?
Right. What does the treatment landscape look like for this group of disorders? I'm interested in how much it's evolved over the last decade, say, but also what's in clinical trials now, or what's just going to enter in the near future that gives you hope, or has you thinking about there might be other options around the next bend?
Yeah, so one, the medical foods have improved tremendously, the taste, the low protein, which is the mainstay for many patients right now. Two, the medications have improved onto the third generation of nitrogen scavengers, so that's improved over time. We do have a phase 3 gene therapy trial in place for OTC. And there are other things coming on the horizon. I'm not really able to talk about them right now, but other ways to either look at or edit the DNA, the RNA, the proteins, or even repurposing drugs. So I think there is a landscape there. I think what's missing at this point is the neuroprotection, right? So if the patient has a hyperammonemic episode, is there something we can give to try to mitigate or minimize the neurologic damage that may come out of that event, especially in the newborns?
Yeah.
Yeah.
Yeah. And do you think that will require it ultimately, like, are the therapeutics going to target the liver and you address the issue there and then it fixes it systemically, or will you need to have a different approach for the brain potentially? I'm just trying to think about the practicalities of
I think
different
you're going to
medicines you
have
can
to
develop.
have a different approach to the brain. It would be similar to maybe a TBI approach and possibly a cocktail
Yeah.
of different medications. Yeah, I think once the patient has a definitive therapy, then, that should be okay. If there's a gene therapy and it's sustainable, it's durable, that will target the liver because the urea cycle is in the liver. But I think, if the patient has hyperammonemia in that moment, you really need to also target the osmotic changes in the brain, the biochemical changes. And again, we can monitor that with the imaging. So we can look at diffusion, which is usually the first thing to change in real time, quickly in the brain after hyperammonemia. So I think neuromonitoring will be the way to go, hopefully at the bedside, hopefully with using less invasive devices. We haven't used our FNIRs at the bedside. That's the next plan for us right now. We have caps of all different sizes down to premature babies. So I think, and also other wearable devices. So there are some companies that are developing devices where you can actually walk around with them, and virtual reality type. We may replace our driving machine with a VR system that also tracks maybe EEG and FNIRs into the same cap. So I think, the other thing we're interested in is biodesign. So the concept of, is there an unmet clinical need here, and how can we use technology to address that? So I do have a biomedical engineer in my group, and so I've tasked her with looking at that to see if there's any way to improve the technology to be able to use it for some of these questions that we have.
Yeah, it makes total sense. Fascinating. If there's one or two areas to close out on that you're really keeping an eye on, whether it's new technologies that you think people should be thinking about if they work in this area or adjacent areas, if it's areas that you think need more attention, that if you could, wave your magic wand and say, if I could have technology X or Y, things would be a lot better. I'm curious what you're thinking about for the future. What are the areas you're most interested in?
So I think, bringing the diagnostics to the clinic more readily and easily without the hang up with insurance, and getting it out there to adults as well. I think one problem we're going to have, and I think we have to flood the literature with articles, is the dual diagnosis. And so if you do a genetic test and you come up with a diagnosis and you feel like that doesn't explain the whole phenotype, and so then you argue or appeal to insurance to say, well, we did the whole exome and it came up with this, but this only explains this part. And, we think there's a second disorder as well. They're probably going to refuse it. They're going to say, why? You already have a diagnosis, you have a positive test. So I think that's going to be difficult. And I think also, with dual diagnosis, gene therapy is not going to be the answer because it will only address one, but not the other condition necessarily. So I think dual diagnosis, adults, and just the whole process from bench to patient. So the basic science to understand the mechanisms and potential therapeutic targets, and the FDA accepting other preclinical models, because there may not be an animal model that's a perfect fit. So cell culture or computational simulations to identify molecular pathways or genetic variants. And then the preclinical studies, the clinical trials, the regulatory approval for treatments, the clinical implementation, and then the real world evidence in diverse populations. I think, a lot of our clinical trials don't have diversity. And so we may not be looking at the whole disease, and that can help refine treatment protocols. And then the iterative improvement. So because precision medicine is evolving and the new discoveries and technologies will refine our understanding of disease mechanism and approach, we have to continually evaluate and refine our strategies based on emerging evidence and patient outcome. So I think, more than ever, this process is collaborative and really is the definition of team science. So I think, everything's going to improve, but we really have to tap people from the different specialties to weigh in and to continue to improve this so that we can really, do precision medicine, which is the long-term goal.
Yeah, absolutely. Well, I thought that was a great note to end on. I wanted to give you an opportunity to just share your website, anything you're looking for, if you're looking for collaborators, if you're hiring, anything like that. Now is the opportunity to shout it out. If people want to get in touch with you, what's the best way to find you?
They can probably just maybe through LinkedIn. Might be the best way to find me. Perfect. Or my email address, agropman@childrensnational.org. Yeah, definitely interested in any bioengineers out there, or anybody who does translational basic research for inborn errors would be great. And also any drug companies. We do have a repurposed drug, a colleague of mine and myself are looking at in mitochondrial diseases. So interesting.
Yeah. Are you that is I think it's such an important and under invested area of drug development, right? We've got these amazing, often proven safe assets that are sitting on the shelf somewhere that could be repurposed in an effective way. You're thinking of partnering with a biotech or drug company to take it through clinical trials, and I guess you could probably do a lot of the preclinical work in your group, right?
Right. So we've done the preclinical, and we can do the phase 1, 2, but this would be a repurposing, but a reformulation as well. So yeah, definitely would, for the definitive phase 3 would need to partner.
Yep, definitely. Well, great. Thank you so much for doing this. I really enjoyed the conversation. I'm sure everybody else listening did as well.
Okay, thanks so much.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.